Hello, BISC1323. This is the beginning of recorded lecture 3-1, and we are still in plant world, but not for much longer. There's just one last topic in this chapter on plant form and physiology that I need to finish up before we can move on to animals. So the topic has to do with transportation, and it's really kind of two topics because there's transportation of water, and then there's transportation of solutes, things like sugars. So let's talk about the transport of water first. So this journey is going to involve the concept of water potential. If you look at this section in the textbook, there are a lot of numbers and it gets kind of mathy. I am not doing a math version of this. We're just going to talk about this qualitatively. All you need to know regarding water potential is that water potential is the potential energy of water, as if that wasn't obvious. But here's the important statement. Water is always going to move from high water potential to lower water potential. This is not the same phenomenon as osmosis, but it is very similar to that. Just know water moves from high water potential to low water potential. Okay, so the journey of water is from the soil, through the roots, up the stems, and into the leaves. It's going to be in the soil because that's where gravity takes it. It needs to go to the leaves because they are the parts of the plant doing photosynthesis, and they need water to do photosynthesis. So, step number one, from the soil to the roots. So, this is going to happen by lowering the water potential in the roots, and the cells of the plant can manipulate their own water potential, create this sort of negative water potential in the root by packing it full of solutes. By putting a bunch of solutes inside of these cells, it makes negative water potential. Water is just going to naturally flow into the root. Importantly, they don't have to spend any energy to do this. This is happening for free because water just naturally moves from high water potential to lower water potential. So increasing concentration of solutes in the root cells makes low water potential. Water naturally is going to move into the roots from the soil. Next, we got to go up the vascular tissue, specifically up the xylem. This happens because of cohesion and adhesion, but mostly adhesion. This is a concept that you may remember from BISC1323, the concept that water sticks to itself, but water also sticks to things around it. It has to do with the hydrophilic nature, all the hydrogen bonds in water. Because water likes to stick to things, if you put water in a thin tube, it's going to be attracted to the walls of the tube and climb up the walls of the tube against the force of gravity. And we learned about the vascular system earlier in this chapter. And it's just a series of tubes. And so, again, without having to spend any energy actively, water is going to be drawn up the walls of the xylem because of this process of adhesion. So up the roots and the stems, because adhesion draws water up the walls of the xylem. The key term's definition of adhesion is the attractive force of water molecules and other molecules. So water sticks to the walls of the xylem. Okay, now the weird part, we have to get this water from the xylem into the leaf tissue, the tissue that's actually doing photosynthesis that needs that water. This is going to be accomplished through a process known as transpiration. So let me read the key term's definition of transpiration. Transpiration is the loss of water vapor to the atmosphere through the stomata, or again, it's labeled here stoma, same thing. Let me read that again. The loss of water vapor to the atmosphere through the stomata. So we went through all this trouble of moving water in through the roots, or into the roots, up the xylem, into the leaves, and then we're just going to get rid of it. We're going to let it exit the plant and evaporate into the atmosphere. Yes, we are going to do that if we're assuming we're plants here. Yes, plants are going to do that because that creates low water potential in the leaves, and that is going to make this water move from the xylem into the leaves where they can use it. This is kind of like, it feels wasteful because you're getting rid of all this water that you spent all this trouble of transporting. And indeed, about 90% of the water that most plants take up from their roots just leaves the plant again through the process of transpiration. But it's worth it because this all happens without having to spend energy, without having to spend ATP. So you're spending water to transport water for free. I'd say that's a pretty good deal. So from stems to the leaves, this occurs through transpiration, as we just defined. It creates negative water potential in the leaves. Water moves from high to low water potential, so it's going to move into these leaves from the xylem. So that was the journey of water. That was the journey through xylem. There's a second half to this involving the phloem, involving the other vascular tissue. So some terminology first. Photosynthesis produces what we can broadly refer to as photosynthates. Sucrose and glucose and other sugars are products of photosynthesis, but the term photosynthate is just more broad, stuff that's made through photosynthesis. Food, important, useful molecules. The places that make photosynthates are called sources, example, leaves, and they're transported to places that need to receive those photosynthates. For example, roots, but it's not just roots that need sugars. They're obviously not doing photosynthesis, but also young parts of the plant that are actively growing really fast. They need to be supplied with energy, and certainly developing seeds need to be given energy as well. And importantly, this can be up or down. This process, to go back to water, this was always from the ground up to the leaves. But this process of moving photosynthates, it can be, from the leaves down to the roots, or it can be from the leaves up to developing seeds, or from the leaves up to young parts of the shoot. This could be in either direction. That being said, the example is showing movement from up to down, but it could be either direction. So here's our process of moving photosynthetes. We call this translocation. So the first step here is moving these photosynthetes from a source cell, parentheses leaf, looks a lot like a leaf to me, from the leaf to the phloem. This is accomplished by active transport. This is a different type of active transport, but this was the example I used back in BISC1323 of something where we're actively spending ATP, we're actively spending energy to pump stuff from one side of the membrane to another. We are actively spending energy here, moving these photosynthetes into the phloem. Journey of photosynthetes, it's called translocation, and step number 1, photosynthetes are moved from source cells to the phloem by active transport, and yes, it costs energy. Now, we need to move these photosynthetes down. This actually plays in to the xylem. There's a reason why xylem is next to phloem. In fact, on all these slides that we saw earlier in this chapter, I know that was for the previous exam, but on all of these things for the previous exam, we always had the xylem and the phloem next to one another as part of these vascular bundles. The reason why they have to be side by side is because of osmosis. Here's another blast from the past from BISC1323. Osmosis is defined in the key terms as the movement of water through a semi-permeable membrane from an area of low solute concentration to an area of higher solute concentration. Well, as it turns out, xylem doesn't have a whole lot of solutes in it. It's not pure 100% water. There are some ions and stuff dissolved in the water in the xylem, but it has pretty low solute concentration. In contrast, in the phloem, there are a lot of solutes, all these sugars that we just actively transported in there. So water is going to naturally move from the xylem to the phloem. So osmosis causes water to move from the nearby xylem to the phloem. And yes, now water potential comes up again. All this water entering the phloem, increases the water potential in the phloem. So now we can finish this journey by moving these photosynthates into the sink cells. In this case, it's a root. Why would they do this? Well, these sink cells have low water potential, as we discussed talking about the transport of water. And this water, along with all the dissolved solutes, are going to move into the sink cells. So the phloem contents, which include the important photosynthates, move to sinks which have a lower water potential. Water always moves, along with all the stuff dissolved in it, from high water potential to lower water potential. Whew, okay. That was kind of a doozy. Again, I didn't want to try to cram this in on the previous exam, but that does bring us to the end of this plant chapter, but not to the end of this lecture. So let's continue on with this lecture, moving on to animals. Now, as we talk about animals, we will definitely talk about animal diversity. There will be a lot of phylogenetic trees in our future. But before we start talking about that, there's kind of a small little chapter here that's just kind of some general introduction to animals as a group. Just looking at some of the features and some other miscellaneous things about animals that will set up those chapters talking about different groups of animals. So some of the traits, animals are multicellular. We're all multicellular. Some animals are microscopic, but all animals are multicellular. We are heterotrophs, which, again, we've seen this many times in the key terms, but it is an organism that consumes organic substances or other organisms for carbon. There are a lot of different ways to be a heterotroph. Some are carnivores. Some are omnivores, some are herbivores, some are parasites. All of these are types of heterotrophy. We see all of this stuff within kingdom Animalia. Most animals have complex tissues. The one exception is going to be the simplest animals, sponges, which we'll see soon enough. But most of them have complicated tissue. Importantly, no cell walls. Plants had cellulose cell walls, and fungi had chitin cell walls, and bacteria had peptidoglycan cell walls. Animals do not have cell walls. Instead, we have this structure called the extracellular matrix. It provides some external protection, but it's squishier, it's more flexible. Animals have what's called a diplontic life cycle. So no more of this nonsense. That was a plant thing. The diplontic life cycle of animals means the multicellular form is diploid. We do go haploid. We do have part of our life cycle where our cells are haploid, but they're never multicellular. It's just a single haploid sperm cell, a single haploid egg cell. We don't have a multicellular haploid like we saw with plants. So this is called a diplontic life cycle. The multicellular form is only diploid. Most animals, compared to fungi and plants, are motile, which is, reading from the key terms, an organism capable of self-locomotion. However, some animals are sessile or sessile, however you want to pronounce that, an organism that is attached to a surface and is not capable of self-locomotion. Bears run around, fish are swimming around, barnacles and stuff, that's not a barnacle, but mussels and barnacles and plenty of other animals are sessile. Animals have complex development. And a fixed body plan. This statement needs some clarification. So when I say a fixed body plan, I mean as part of our development, which follows a pretty strict process, which I'll talk about later in this chapter, it brings our bodies to a fixed state. This bear, for example, goes through development that says 2 ears, one on either side, 2 eyes, one on either side, 2 nostrils, one on either side, one heart, and all the art. This is all programmed in to how its development is supposed to be. Compare that to plants or to fungi. There's no strict rule that says, okay, these trees have to have exactly 20 branches, and half of them need to be pointing one way and half of them need to point the other way, and each one of those branches has this many leaves. There's some randomness to this, how long the branches there are and how many there are and how many leaves on each branch or whatever. This is determined by a lot of environmental things, and fungi are the same way, but animals are much more strict than these other groups of multicellular organisms in having this program to have a very specific body plan. Animals have sexual reproduction. So, yep, egg plus sperm equals zygote. We've seen that before. But there are also some animals that are capable of asexual reproduction. So, yep, here's the egg plus sperm equals zygote. And here's a sea star undergoing asexual reproduction. So, yeah, we see either of these throughout Animalia. Okay, now that we've got some just generic traits out of the way, I want to talk more about this picture, this embryonic development. So I'm not going to go through all of embryonic development, but these first few steps are important to understand because we're going to use some of the terms later to classify different groups of animals. So this process starts with a zygote, egg plus sperm equals zygote. We have this single-celled diploid zygote. And the zygote is going to go through several rounds of cell division. But there's something weird about these cell divisions. Typically with a cell division, the cell grows in size and then splits in half. But this zygote is a massive cell, all things considered, because the egg is a massive cell, all things considered. These first few rounds of division that it does involve it splitting in two without growing in size. So we end up with an 8-cell stage embryo that is the same size as the 1-cell zygote. This kind of dividing without growing is called cleavage, and it's going to continue to happen early during development. So the zygote divides without growing in size several times. This process is called cleavage. It goes from 1 cell to 2 to 4 to 8, and you know how doubling works. Eventually, these divisions produce a hollow sphere of cells, which we importantly call a blastula. So we're looking at a cross-section so we can see that it's hollow on the inside, but this sphere of cells is called a blastula. Now, we know it's not going to stay a sphere. If you think about your own adult body, you are not really sphere-like. If anything, you are tube-like. With a mouth at one end and anus at the other end, we're tubes. So we need to somehow go from sphere mode to tube mode. The way we do that is by making an indentation in this sphere that is eventually going to punch out to the other side, and then we're going to have our tube with a mouth at one end and an anus at the other end. This process is called gastrulation. The indentation that is made in the blastula is called a blastopore. I'm being careful with my language here because some of these are easy to mix up with one another. Blastula is the sphere, blastopore is the indentation. The process is called gastrulation. I've got it outlined here. Gastrulation occurs as an indentation in the blastula called a blastopore forms. And this creates our digestive tract, our gastrointestinal tract, which is why it's called gastrulation. And this also results in different layers of cells. We'll explore this more later in this chapter, but there are going to be cells on the inside of the tube and cells on the outside of the tube, and we'll see cell layers soon enough. But that's an important consequence of this process. So I'm going to stop there, as far as development goes. There are many, many more steps, but I think this is the most important part to focus on. Now, this development is very similar in very diverse groups of animals if you were to continue on past these stages. What do I mean by very similar? Well, early embryos of diverse animals look very similar to one another. try to guess what these 6 animals are going to grow up into. I'd be surprised if you got all of them right, because they kind of all look the same. However, as embryonic development continues, there are differences that accumulate. Maybe you can guess that one of these looks pretty fish-like, and these look a little bit more complicated, but early on, almost identical embryos, differences come in, and then, okay, now we see some pretty big differences. Looks like a beak there, and if you want spoilers, fish, salamander, tortoise, chicken, rabbit, human, but early on, they all looked the same. This is because we're all using even though we're very different, humans very distantly related from, fish and birds and reptiles, we're all using a very similar program for development that, again, starts off the same and then eventually starts to make some changes. This is due to what are called homeobox genes, or Hox genes. The Hox genes are genes that all vertebrates have, almost all animals have, that control things like limb development and spine development and head development, and all these different vertebrates have these same genes, which is why they're so similar. So this is kind of hard to summarize. Just know that these Hox genes are responsible for the similar development. These Hox genes are in virtually all animals, and yeah, it just refers to a bunch of like big control genes that are responsible for controlling development. All right, let's move on to some topics that are maybe a little less abstract, and talk about features that animals have that can be used to classify them. So let's start by talking about something simple: symmetry. There are 3 main types of symmetry that we see across kingdom Animalia. One of these is no symmetry at all, asymmetry or not symmetrical, and we're going to see this when we look at sponges. But we don't have to get very far before we start to see radial symmetry. Radial symmetry is, here's a sea anemone. Let me read the key terms definition of radial symmetry. A type of symmetry where multiple planes of symmetry, with body parts arranged around a central disc. So any sort of plane that you draw through the middle is going to make mirror images on either side. And then our third type of symmetry, the most common one by and far, is bilateral symmetry. We see it here in this goat, but you can see it in your own body. Bilateral symmetry is where there's only one plane that you could draw through the middle of the animal to make mirror halves. The key term says a type of symmetry in which there's only one plane of symmetry, so the left and right halves of an animal are mirror images. A couple of related concepts when we are talking about bilateral symmetry, and again, this is the most common type of symmetry, so we'll see this a lot. There are 4 important terms: anterior, posterior, dorsal, and ventral. Anterior refers to the front of the animal, the end with the mouth, because again, we are just basically tubes. Posterior refers to the opposite of the anterior, where the anus points. Dorsal refers to the back, where there's the spine, if we're talking about something with a spine. And then ventral is opposite of dorsal, towards the stomach. These are terms that we see in bilaterally symmetrical animals. Anterior is front, posterior is rear, dorsal is toward the back, ventral is toward the stomach. Okay, another way to classify or categorize animals goes back to this thing when I was talking about cell layers being created here. So remember, our bodies are kind of just tubes. If you were to cut a cross section of this tube, you see what I mean. There's the interior cell layer lining a digestive tract, and there's an outer cell layer making up your skin. Now, the simplest way to have cell layers is what we're looking at here, an animal that is a diploblast. Di means 2, we've seen that before a few times. Diploblastic animals have 2 cell layers. The interior layer of cells is called endoderm. The exterior layer of cells is called ectoderm, and in between the two is this non-living layer that's used for different things depending on what you're talking about, but it's not cells. There are only two layers of cells in a diploblastic animal. Diploblastic animals have two cell layers. Endoderm is interior, ectoderm is exterior. Now, we're not going to see too many diploblasts. What we're going to see a lot more of are triploblasts, including ourselves, if you're curious. Triploblasts have three cell layers, as you could guess from the name. The outer layer is still called the ectoderm. The inner layer is still called the endoderm, but instead of having non-living jelly in the middle, there's a middle layer of cells called the mesoderm. So triploblastic animals have three cell layers: endoderm, mesoderm in the middle, and then ectoderm on the exterior. Now, among these triploblasts, there are three groups based on an additional body cavity. Acoelomates, eucoelomates, sometimes just called coelomates, and pseudocoelomates. These are all defined in the key terms, but again, a picture is worth 1,000 words. What do we look at? Here is an acoelomate. Acoelomates, again, it's just a cross section. These are some examples here, but pay attention to the cross section. An acoelomate is a triploblastic animal that doesn't have any extra layers. You've got the ectoderm, the mesoderm, the endoderm, you have the digestive cavity, but there are no other cavities, there are no other structures, nothing else going on. Coelomates, or eucoelomates, we're using them interchangeably here, have ectoderm, mesoderm, endoderm, but then they have this body cavity called a coelom. It's not made of cells, it's an empty cavity. They use it for lots of different functions. This coelom is located within the mesoderm. Notice how this is completely surrounded by the green mesodermal layer. The coelom body cavity found in eucoelomates is within the mesoderm. And finally, pseudocoelomates still have those three cell layers. They have an extra cavity called a pseudocoelom. It is not within the mesoderm. Notice how this pseudocoelom is located in between the mesoderm and the endoderm. Again, the cavity is used for lots of different things. We'll talk about all these different types of worms eventually in the coming chapter, but these are different types of body cavities that we see within triploblasts. Again, all of them in the key terms, but this is a great figure to look at to visually understand the differences between these three. Okay, there is one more distinction here. Among animals with bilateral symmetry and a triploblastic body plan, there are two distinct groups. They are called protostomes and deuterostomes. Now, there's a lot of crazy stuff going on here, but really we could focus on just one important difference between protostomes and deuterostomes. So here's our eight cell stage. Here's the process of gastrulation. There's the blastopore, that indentation. In protostomes, the initial indentation is the mouth, and it eventually extends through to form the anus. But the first part that forms is the part that is going to be the mouth. In deuterostomes, it's the opposite. The initial indentation is going to eventually be the anus. It's going to extend out and form a mouth on the other side. So protostomes, mouth first, deuterostomes, butt first. That's the biggest difference. And this feels like maybe it's way too fiddly of a distinction that doesn't matter, but trust me, when we look at phylogenetic trees, not everything we talk about is going to be a clade. Not every traditional group is going to be a monophyletic group. But protostome is a clade, and deuterostome is a clade, which is why it's worth introducing now. So protostome, the key term says a lot. The key term says a group of animals in which the blastopore develops into the mouth with the second opening developing into the anus. So mouth, then anus. Deuterostome, a group of animals in which the blastopore develops into the anus with the second opening developing into the mouth. Deuterostomes are anus and then mouth. Okay. There's one more section in this animal diversity chapter, but I think that's going to be left to the next lecture. This is not the end of animal diversity, but this is where we're coming to an end with recorded lecture 3.1.